Hello everyone, welcome to my channel. Today we are going to learn about microsporogenesis. In the previous class, we have learned about the structure of microsporangium. So this is the continuation of the last class. Microsporogenesis. Genesis means formation. That is the formation of microspore. Microspore is the male gametes, male gametophyte, microspore. From the microspore mother cell. From the microspore mother cell, which is otherwise called as pollen mother cell. Pollen mother cell, or the other name which is given for this one is meiocyte. The microspore mother cell, if you see here, it is a diploid cell, right? This is the microspore mother cell which undergoes the first reduction division, meiosis 1, and forms two haploid cells, right? Again it will undergo the second reduction division, meiosis 2. Then here it forms a microspore which is tetrahedral in structure. All the microspores will be arranged in a cluster of 4 cells, right? Like here, cluster of 4 cells. This is the 3D structure of a microspore. 1, 2, 3. 3 will be visible from the front side, and then one more will be there at the back side. That means total 4. So it is 4 cells together, they are forming a cluster, and these are called as in tetrahedral. And in other species, they can be in different forms, right? This microspore formation, they can be in different forms. Let us see what are they. Okay. If the 4 haploid cells are arranged in a cluster in this manner, then this condition is called as isobilateral. Isobilateral. Next, if the 4 haploid cells are arranged in this manner, then this condition is called as linear. One more condition here exists, which is very rare, where the haploid cells are arranged one above the other, and then 2 haploid cells will be arranged one above the other, and the rest of the 2 will be arranged adjacent to each other, here like this. This condition is called as decussate. These are the different forms of the arrangement of the 4 cells, that is 4 microspores, 1, 2, 3, and again one more will be at the back. This is the first one, microspore, tetrahedral structure we say. This kind of arrangement we call it as isobilateral, and this type of arrangement where one upon the other the haploid structures are arranged, haploid cells are arranged, called as linear, and the last one is decussate. Right? Now this microspore, this is the very common one. Tetrad is the most common. Okay, these microspores are formed and are arranged in clusters. Now as the anthers mature and dehydrate, the microspores will dissociate from each other. Now they are forming a cluster and they are attached with one another, and they will dissociate with each other and form single cells. So when the anther is mature, these microspores will dissociate from each other and develop into pollen grains. These are the haploid pollen grains. In the previous class, I have mentioned that because the pores or the microspores are attached with each other, they will be dehiscized, or they will be separated only with the help of an enzyme called as catalase. There is a small correction here. It's not catalase, it is calase. Now this is the enzyme calase which is released from the tapetum, right? Okay, because of this enzyme which is released from the tapetum, the walls of the microspores gets ruptured and the pollens are released out. That means they get dehiscized, forming 4 different pollens that are haploid in nature. Now let us study in detail about the structure of pollen grains. Pollen grains represents the male gametes or male gametophytes. They are generally spherical in shape, which has a width of nearly 25 to 50 micrometers in diameter. Pollen grains represents the male gametophytes and they are spherical in shape, which has a diameter of about 25 to 50 micrometers. These pollen grains are haploid structures. Now let us see how the structure is. This is the structure of a pollen grain, which is generally two-layered. The first layer where we have formed the ridges and the smooth, two different type we can see in the first layer. First layer here, this part is called as exine, which is the outermost layer, and which is the hardest layer, we can say hard outer layer. Hard outer layer, and this part contains a substance called as sporopollenin. Because of the presence of this sporopollenin, it is very hard in structure, and also because of this, it can withstand the high temperatures and also the strong acids and alkali. No enzyme till now is responsible or can degrade the sporopollenin. Now here in the outermost structure only, the region which between these rough surfaces is called as germ pores, right? Now this germ pore, which is nothing but a gap between these two exine, and here the sporopollenin is not there. The sporopollenin is not present in these germ pores. The second layer here is the intine, which I have shown with the red color. This is the second layer called as intine. This layer is thin and continuous layer, which is a thin layer, and also it forms a continuous layer, and it is made up of cellulose and pectin. Now here inside is the cytoplasm. This is the cytoplasm. The cytoplasm of this pollen is surrounded by a plasma membrane. So that means two most important structures, the outermost layer which is exine and the innermost layer is the intine. Okay. Now this is the general structure. When the pollen grain gets matured, it will contain two cells. Let me show here. When the pollen grain is mature, it contains two cells, vegetative cell and the generative cell. Vegetative cell is bigger than compared to the generative cells, and it has abundant food reserve materials. It has food reserve materials, right? And also has an irregularly shaped nucleus. Here, this is the irregular shaped nucleus of vegetative cells. Now, in case of generative cells, these are the small than compared to vegetative cells, and also it will float in cytoplasm of the vegetative cell. This is what is here the cytoplasm here. It is very small, spindle shape, and is having a dense cytoplasm and a nucleus. Now this stage is called as two-celled stage, where the 60% of angiosperms shed the pollen grains, whereas the 40% of them, they will enter a three-celled stage. So this stage, particularly where the pollen grain after maturity forms two cells, is called as a two-celled stage, upon which the 60% of the angiosperms, the pollen grains are shed, right? Pollen grains are shed, and here the generative cell will divide again mitotically to give rise to the two male gametes. Okay, two male gametes. Out of the 40%, they will enter the three-celled stage. Till here we have studied about the structure of pollen grains and how they are matured. Now let us see what are some disadvantages as well as advantages with the pollen grains. Now if we see pollen grains, I'll write here, they cause allergies and bronchial afflictions. So what is the main disadvantage of these pollen grains? They cause allergies and also the bronchial diseases like respiratory disorders, okay, like called as asthma and bronchitis. Like example, the common example can be taken as carrot grass, which we otherwise called as parthenium. This came to India, actually this was not present here before. This came to India as a seed contaminant. with imported wheat and caused the pollen allergy. What is the advantage of these pollen grains? Now, pollen grains are rich in nutrients and hence they can be used as pollen tablets, as food supplements. These pollen tablets or syrups, these are used for consumption to increase the performance, especially in case of athletes, and are also given to the race horses. These are the disadvantages and advantages. Let us quick revise. The pollen grains are rich in nutrients and hence these has been used as like pollen tablets or syrups, right? And these pollen products are used in the form for athletes and also given to the race horses to increase their performance. Now let us talk about the viability of pollen grains. Okay, what is viability? Viability means the ability to live. So once the pollen grains are shed, they have to land on the stigma before they lose their viability, right? Now this period of for which the pollen grains remain viable will depend on the species and also upon the external temperatures and humidity, right? Next, like example, one best example, if you are taking cereals, like example rice, wheat, so in this case, the like example in case of cereals, rice, wheat, pollen grains will lose their viability within the 30 minutes of their release. So that means they have to reach the stigma before 30 minutes or else they will lose their viability. Whereas in case of some members like rosaceae, Leguminaceae and Solanaceae, they will maintain the viability for months. Now here it is also possible to store the pollen grains in large number of species for months, like or for years also. We can store the pollen grains for years. And how is it possible? In liquid nitrogen by maintaining a temperature of minus 196 degrees Celsius. Such stored pollen grains are called as pollen banks. In the same way, how the seeds are stored and called as seed banks, in the same way here also, the pollens are also stored and can be used for years as pollen banks. Now this will help in the crop breeding programs. This was about the structure of pollen grains and microsporogenesis as well. In the next class, we'll study about the megaspores and megasporogenesis. I hope you understood. Thank you.